Hi everyone. Welcome to this workshop session within the LRIG meeting, Translating Ideas into Therapies. My name is Dave Madge and I represent WuXi AppTec in Europe, supporting our many clients in this region. For those of you that don't know us, we provide a broad portfolio of research, development and manufacturing services, enabling companies of all shapes and sizes to discover and develop groundbreaking therapies and deliver benefits to patients. We work across all therapeutic areas and support small molecule, peptide, and oligonucleotide therapeutics, as well as cell and gene therapy and medical devices. Today, our open access platform is enabling more than 4,000 collaborators globally. It's a great pleasure to be here today to introduce this session on target identification and validation. Like you, we place a great value in connecting with scientists working in areas related to our own, to hear about the latest developments in technologies and their application in drug discovery. So we're very grateful to the LRIG team for this opportunity. In our workshop today, we'll first discuss strategies and technologies for identification of new molecular targets, in particular using gene manipulation technologies. My colleague, Dr. Baiqiu Wang, will present this section. Thereafter, we'll focus on target validation, and Dr. Shelley Zhong will lead this discussion. Both Baiqiu and Shelley are from HD Biosciences, the business unit within the WuXi AppTec family. Both are based in Shanghai. After these discussions, we'll have a little time for a question and answer session, but we'll try and leave you 5 minutes to grab a coffee before the keynote speakers. A few words of introduction then to our first session. We're going to cover CRISPR library screening for target identification and mechanism of action confirmation. The speaker, as I mentioned, is Dr. Baiqiu Wang. Baiqiu has more than 14 years experience of drug development working at HD Biosciences, focusing on early stage target discovery and validation, assay development, screening, biomarker discovery, and new technology development. Prior to joining HDB, Baiqiu had research training at Brigham and Women's Hospital in Boston and received her medical degree and her PhD in medical genetics at Harbin Medical University. I hope you enjoy the discussion. Please remember to ask questions. And of course, you are able to follow up with us using the contact details that will appear at the end of the slides, both speakers. You can submit questions using the Q&A button at the bottom of the Zoom screen. Baiqiu, over to you.
Good morning, everybody. My name is Baiqiu. I am very happy today to join this online conference and introduce the CRISPR library screen platform from HDB. So this CRISPR library screen platform has been up and running at HDB for several years now. The major purpose of this platform is to help our clients to identify some new target that can be used to develop some new treatment. And on top of that, we can also utilize this platform to help our client understand better the drug-resistant mechanism. Therefore, we may develop some combination treatment strategy. And after identifying some of the hits from the library screen, we usually move on to further confirmation of those identified hits. And in doing so, we may also identify some potential biomarker that can be used for tailored treatment strategy. So let's take a look at the summary of this CRISPR library screen capability. So first of all is the CRISPR library. What kind of library do we have here? We have the CRISPR knockout library, and we also have the CRISPR-I and CRISPR-A library. So for CRISPR knockout library, we usually have the library targeting at the whole genome. In the meantime, there are also library that has focused functions. For example, the epigenetics focused library, the cancer metabolism, or even the ubiquitin library. And CRISPR-A and CRISPR-I library are those library that focusing on the transcriptional regulation. So in terms of the CRISPR screening types, we have several options here. First is the synthetic lethal screen. This synthetic lethal screen can be carried out through particularly select compound treatment, and then followed by library screen. And another way is to use engineered cell line, where you overexpress, knock out or knock down your target of interest, and then perform the library screen to identify the partner which you can use the full combination study further. So we also have the positive screen, where we utilize high compound selection pressure and then identify some hits for the resistant mechanism. And for negative screen here, we usually use much less selection pressure and then identify the drug sensitizer here. And of course, we always include the DMSO control for the regular CRISPR library screen. So let's take a look at this internal validation effort at the very beginning of setting up this system. So to validate the system at HDB, we choose lung cancer cell line H1975, which has several EGFR mutation. And for the compound, we actually select Plasmid, which is an EGFR HER2 inhibitor. And after a treatment of this compound for 7 or 15 days, as you can show here in this figure, we can observe a very significant cell survival as opposed to the naive cell line. So when the compound treatment is completed, we harvest the sample and get the DNA sample ready and then send out for sequencing. So this is the analyzed result here. So showed quite significant curve shift between the 7-day treatment group and the control group. So those shifts represent a depletion of a certain subset of the GET-RNA. And we also observe certain group of GET-RNA enriched after the drug selection. This is analyzed the result that indicate certain GET-RNA enrichment actually are greater than 10-fold, 30-fold, or even 50-fold here. So the right line actually showed the GET-RNA enrichment over 30-fold. So those identified high-ranking candidate, we can see there are some of those hits already well established to be closely associated with drug resistance. In the meantime, we also identify certain new targets that definitely worth further investigation. Now let's take a look at the 3-screen-in-1 drug modifier platform that currently running at HDB. So for this slice, we would like to introduce the general process. On the right is the experimental flowchart. So you can see we transduce the Cas9 stable cell line with the CRISPR library first, and then we do the pure mesene selection. Followed by that, we remove the pure mesene and split the cells into 3 groups. The first is the positive screen group, where we use high compound concentration, usually is IC90. And then in the middle is the low compound concentration group, where we usually use IC20, which is, we also call it the negative screen. And then is the DMSO control group. So on the left is a general flow chart. You can see we usually start with the early stage parental line characterization, and then followed by that is a stable cell line generation and characterization. And then we prepare the GET-RNA lentivirus and make sure we have the various titration. And then we expand the cells and start the screen. Following the screening, we will extract the gut DNA and then do the barcode PCR amplification. And then it's the deep sequencing and data analysis. So these are some representative data. So for the parental line characterization, for example, here we have some routine, seeding density optimization of antibiotics titration. The very important data here is the Western blot in the middle that shows the overexpression of Cas9 protein. But not only just confirm the Cas9 overexpression by Western blot, we also need to confirm that Cas9 is functioning properly. We usually do that through a fluorescence-based system where we can confirm that the Cas9 protein is functioning. Another very key step is to determine the compound concentration. So, like I mentioned in the previous slides, we usually choose IC90 for the high compound concentration, and then the IC20 for the low compound treatment. And on top of that, the GAG-RNA library titration is also very key. We need to determine how much virus we need to use for certain amount of cells to eventually achieve a transduction MOI of 0.5. And then before we do the data analysis, there are a few key steps of quality control. So the DNA extraction and barcoding application, the PCR test, those are very basic quality control. Another important QC in the middle the data shows is to confirm the data consistency between different replicate. So this is very important for the final data analysis. And on the right, the curve shows another important step of the quality control is to confirm a certain group of essential genes depletion in this screening. This is the last step to conclude that we have a good quality control before we moving on to data analysis. And this is a slide that shows some of the hits identified from the library screen actually correlate very well with the known biological functional test of the selected compound. For example, here, from the library screen, we identify this drug target X that is very much related to the drug resistance development. And on the left is actually a cell line that resistant to this compound. We generated, and then we observe the target X expression was downregulated in this compound resistant cell line. In addition, we also have a separate screening, which is sgRNA library screen, use the same compound. We also identify this target X as a hit that's related to the drug resistant development. So all these studies correlate very well with each other, and to also confirm that target X is a very good potential hits for further studies. So this is another case that shows a typical drug-resistant mechanistic investigation where we use library screen to identify potential hits. And then on the right, we also have the CRISPR knockout cell line where you independently confirm that for certain target, for example, target N, if you knock it out, it actually confirms resistant to the drug treatment. But for other target, for example, target M here, if you knock it out, it actually enhances the drug sensitivity. So CRISPR-I, like I mentioned previously in this presentation, is a screening that's targeting at transcription. It worked through silencing the transcription. So for this type of screen, the GetRNA and Cas9 complex target at certain DNA element that's complementary to the GetRNA and therefore block the transcription. So even though the principle is different as opposed to the regular CRISPR knockout library, this CRISPR-I screening, the process is pretty much the same as the CRISPR knockout screen. So we usually use the same Western blot to confirm the overexpression of the enzyme deficient Cas9 and use the fluorescence system to confirm that this Cas9 can definitely block the transcription. And then in the middle, also, we need to verify the data consistency between different replicate. And followed by that is the final analyze the result to identify potential drug sensitizer as well as desensitizer. So this is my last slide. I would like to point out that we have a current ongoing effort to put together a group of cell line, around 50 cell line here, covering different indication, where we already generated a Cas9 overexpressing line. So this will help you to skip a step in your library screen preparation, therefore save time for the whole screening campaign. So we have a different stage, for the stable cell line generation, but I think by the end of this year, we should have all 50 lines up and running. So if you choose to use the cell line on this lift, you will definitely save time. All right, that will be the last slides for my presentation. Again, thank you very much for your time. And I will answer some questions later on, but we also have the email contact information here. If you have any further questions down the line, feel free to reach out to us. Thank you very much.
Thanks very much for that, Baichu. Please do note, everybody, that you can submit questions, and at the bottom of your screen, we will, as Baichu mentioned, have a Q&A session at the end of this discussion. Our next speaker is Dr. Shelley Zhong, also from HD Biosciences. Shelley is going to dive a little more deeply into the target validation at HDB, target validation platform at HDB. Shelley has more than 15 years experience of cancer research, with the last 5 years at HD Biosciences, leading the exploratory biology team. This team has a focus on early target discovery and validation. And in this role, Shelley has built up the advanced gene editing platform, extended CRISPR to primary immune cells, as well as developing technologies to support new modalities such as PROTAC, TCR, CAR-T, oligonucleotide therapeutics, and antibody-oligonucleotide. Shelley received her medical degree from Peking University Medical School and a PhD degree in pharmacology at UPenn. Over to you, Shelley.
Good morning, everyone. This is Shelley Fang Zhang, leading exploratory biology team at HDB Wuxi AppTec. Today it's my pleasure to introduce to you the comprehensive target discovery and validation platform at HDB Wuxi AppTec. So we have an AGM, short for Advanced Genomic Manipulation Platform, that's built around CRISPR technology. And we provide comprehensive phenotypic functional and assay solutions encompassing wide choices disease models. We also have over 1,000 cancer cell line banks that can be utilized for target cells options. We also have mouse genetics and in vivo gene editing platforms. So all this give you integrated platform to support your novel target discovery and validation. So our HTB Advanced Gene Manipulation AGM platforms is having all kinds of innovative technologies, know-how, and multiple delivery systems for gene manipulation, including also comprehensive options from cellular, tissue to in vivo. It provides valuable tools and solutions for in-depth mechanistic studies and proof of concept evaluation. Our CRISPR-centered AGM platform can give you solutions for multiple cell line generation solutions, including knockout, knock-in, overexpression, knockdown, and particularly for overexpression knockdown, it can be constitutive or DOX-inducible. We also have a guide library screen, as just covered by Dr. Wang. So this can help you identify synthetic lethality genes, drug modifier for sensitivity resistance, and also E3 ligase. So these are for molecular glue mechanisms. We have also extended our CRISPR technology, our AGM, to primary cells, including primary immune cells, as well as iPSCs. And CRISPR technology has also been upgraded to base editing and prime editing for better precision and versatility. We provide comprehensive phenotypic assays and models, including just, for example, here, high content and IQ, and also PROTAC target protein degradation platforms, including high-bit knock-in, DTAG, and smash knock-in. So all these leading edge capabilities can support target discovery, validation, and new modalities. So the library screen is for novel target discovery, such as genes for synthetic lethality of drug modifiers, which has just been comprehensively elaborated by Dr. Wang. And this is just a case study showing the drug-resistant sensitive target HIS validated by knockout study. So for our CRISPR, we can do multiple target knockout. So this is an example showing a single or double target knockout in cancer cells. And it's actually, you can do 3 target knockout in either sequential manner or simultaneously, depending on the crosstalk of these targets. The CRISPR-Cas9 technology proved to be efficient, effective approach, particular for multi-target manipulation. And we can also perform CRISPR-based knock-in to validate drug resistance studies and also sensitivities. So this is just to show that CRISPR knock-in of mutation can produce IC50 shift, as demonstrated here. So this just validates these mutations functioning in drug sensitivity or resistance. And CRISPR base editing is an upgraded technology of conventional CRISPR, and it provides more precise genetic manipulation on a single base pair cases. And here it's showing a high percentage mutation for A to G or C to T confirmed by NGS. And CRISPR prime editing as a more advanced technology. It enables higher precision, efficiency, and versatility. And here is the example showing that we have successfully inserted his tag into endogenous HAG3 locus by prime editing. Again, this is standard sequencing results. And we provide a comprehensive assay format and technologies to support target validation. You can see it encompasses biochemical assays, radiometric assays, cell-based target assays, so such as reporter assays, and also Western blots using ECI as well as LIQUOL. And we also have JAS that has a more quantitative for smaller amount of samples. And also we provide high content multiplexing assays in a quantitative high throughput mode. Phenotypic functional assays for primary cells and as well as engineered cell lines, and including various technical platforms and multiple therapeutic focuses. Also biochemical assays and mass spec. And we also have multimodal plate readers, just listed here. And one example, Luminex 3D, up to 500 multiplexed barcoding readings, MSD and LIQUOL Odyssey. So for all these multiple solutions, as well as liquid handling systems as listed here. At HTB, we have 1,000 tumor cell bank with extensive cell type coverage, including 18 different tumor disease models and very wide geographic diversity, and also provide internal genomic data for over 300 cell lines. We also provide complicated, complex assay platform solutions that's listed here. We have successfully extended CRISPR platform to target manipulation in very tough non-proliferating primary immune cells, including mouse and human primary T cells, human monocytes, dendritic cells, M1, M2 macrophages. And these CRISPR-based knockout novel targets have been validated by FACS or standard sequencing. So the efficiency is over 50%. And the short time window enables multiple downstream functional studies like RT-PCR ELISA for cytokines in different primary immune cells. So this just opens up horizon and explorations in the very tough field immunology. So we can interrogate novel targets in immune cells. At HTB, we also provide comprehensive in vitro immunoassay platforms, including immune cell activation for multiple kinds of immune cells, PBMC T cells, NK, monocytes, etc., and immune cell co-culture assays like mixed lymphocyte reactions for assessing T cell activation, M2 T cell co-culture, MDSC T cell co-culture, Treg T cell co-culture for evaluating different kinds of immune cells functionally, as well as immune and tumor cell co-cultures for novel target discovery and validations, as well as immune cell polarizations, so like biomarker profilings and phagocytosis for macrophages. We have developed PROTAC platform at HTB, so this has been extensively utilized, applied for multiple novel targets across multiple kinds of cancer cells. And we also extended this PROTAC platform to primary immune cells. We have very short turnover time, around 1 week, ensures sufficient time for downstream functional assessment. So this is very advantageous for non-proliferating primary immune cells. And the high delivery efficiency ensures excellent assay window, over 100-fold, in the case of step 3. And this in the pooled primary cells shows stability up to 20 days. And this provides options for 384 well-played HTS capability and providing solutions for novel and druggable targets in tough primary immune cells. So here, example of knock-in hybrid in step 3, well-established targets in primary T cells and its functions in inflammation diseases. And we have used a reference compound of PROTAC and showing a degradation step 3 in primary T cells with IC50 well correlative to reports. So this is showing, this PROTAC capability in primary immune cells, helping novel target validation and also screening capability in primary immune cells. We also developed TCR-T CAR-T new modality platforms and demonstrated over 90% high efficiency delivery. So this is advantages, for the downstream processing from the clinical point of view. And TCR-T CAR-T functional validation is performed by co-culturing with MHC or surface antigen overexpressing target cells, followed by cytokine ELISA detections, as you can see the dose curve shown here. We also have developed new modalities such as antibody-oligonucleotides, AOC. This is including rational strategic assay design to efficiently validate siRNA targets and surface receptors for siRNA antibody or ligand conjugates by parallel target cells versus control cells, mRNA, as well as protein expression profiling. We have done comprehensive validations, including multiple layers of optimizations to ensure very high efficient siRNA screenings by RT-PCR as well as ICW. And rational workflow design for target cell surface receptor determination by RT-PCR followed by FACS confirmation for receptor target surface localization. So this is just the showcasing our comprehensive siRNA screening assay optimizations, including transfection reagent, knockdown efficiency, comparison of multiple transfection efficiency reagent to select the most efficient and with the minimized cytotox effect, and also transfection reagent volume time point determination, and housekeeping gene optimization for multiple housekeeping genes across different cell lines. Also, different lysis kit validation by RT-PCR to select the best options that has the best knockdown efficiency. So all these comprehensive validation led to very efficient screening of assays, both the primary screens with 2 dose points as shown here, and followed by the potent hits confirmation for IC50 assessment by a full dose curve assessment. And also parallel with in-cell Western validation in 384 format by LICOR for high throughput Assay screening or compound screening. So this is just showing the Assay screening. You can see the dose response very nicely, and quantitatively, you can also assess IC50s. So for the target cell surface receptor determination, this is just showing the mRNA expression profiling across multiple targets as well as control cells, so we can determine the most promising candidates to confirm by facts. So this is very important for the conjugation by the antibody. Besides AOC, we also have established ASO platform. So this is carried out by the first step setup and optimization for ASO screening by RT-PCR. So including genetic versus transfection approach and time point determination, etc. in human mouse cells, and ASO screening and hit confirmation in both human and mouse cells within parallel cytotox evaluation. And the hits were further confirmed functionally by RT-PCR Western SW with functional validation in human mouse primary cells for enzymatic and cytokine studies. We also have set up new tech development for barcoding our cancer cell panel. So this, we can have pooled 10 to 20 cell lines for drug screening together to greatly boost the drug screening efficiency. And it's also applicable for tumor heterogeneity studies. We expand the CRISPR barcoding to 300 to 400 frequently used cancer cell lines with 2 compound validation. So HTB growing comprehensive target discovery validation capabilities propelled by various new technologies provide solutions for drug R&D across multiple disease areas, as well as new modalities, including PROTAC, TBD, CAR-T, TCR-T, AOC, ASO oligos. And also we have technologies and platform for tumor cell panel barcoding and base editing, prime editing, and CRISPR extended to primary immune cells, iPSCs. So overall, all these provide an integrated comprehensive platform to help provide solutions for target discovery and validation. And here is the contact information for myself. You can email me for any of your science-related questions, or email our business contact Dave for any business-related queries. Again, not to emphasize enough, our mission is to improve health, making difference. Together we deliver. Thank you very much, and welcome your questions in the following sessions. Thank you.
Many thanks, Baichu and Shelley, for these presentations and discussing the team's capabilities with us. We have a little time for follow-up questions, so please do feel free to submit them via the Q&A function. If we don't have time to answer all your questions now, we'll follow up with you directly after this event. So let me get the ball rolling with respect to some questions. Let me start by asking Baichu to talk a little bit about how CRISPR library screening helps us understand the likely resistance generation from targets that are identified during the screening process.
Okay, so the drug resistance is actually a very common encountered issue, and We have quite experienced team to help, design all the experimental plan to handle that. Like I mentioned in my presentation, we usually start by selection a appropriate compound treatment condition. And then we can subject the Cas9 overexpressing cell line to the GetRNA library various transduction. So followed by that will be the compound treatment at the condition that we selected. And then, you collect the surviving cells and then get the deep sequencing data for further analysis. So usually this type of screen will review quite a few potential targets that actually closely related to drug-resistant development. So from there, once you complete the library screen, you can choose further validation. For example, you can generate some resistant cell line or knock out your target of interest to confirm and validate the biological functions. So that's just a very general process for handling a project like that.
Okay, thanks. Thanks, Baichu. Yeah, a general question to both of you, Baichu and Shelley, from one of our visitors. We've both spoken quite a lot about CRISPR as a technology and portrayed many different situations in which CRISPR is the answer to identifying and validating targets. But what do you both feel are the limitations of the technology? What are the areas where CRISPR is not useful, or the areas where we have to be more cautious about interpreting the results that we see? Shelley, would you like to have a go at that first? I think you're on mute, Shelley.
Sorry, so now you can hear me?
Sure.
Thank you, that's a very good question. So I wanted to say that really CRISPR is a very powerful tool. You can have a high efficient knockout that's validated by sequencing and Western blot, and you can see phenotype if you're lucky. But sometimes there are caveats. So one of the things is the CRISPR knockout generation, if you want to generate CRISPR knockout cell lines, it takes time. Even if you do electroporation, sometimes you need some time, little bit lengthy of time to actually generate that knockout cell line, in a particular cell line, cancer cell line, for example. And during that length time of generation, there might be possibly evolution of compensatory mechanisms evolved. So that can blunt your response. For example, this target might be essential, but once it's knocked out, after all these times that you get this cell line generated in the end, the cells were not killed. This was not because this is not essential gene, but because during this time course, there has involved compensation mechanism pathway signalings that can play a compensatory role. So that has been actually realized and by multiple clients' projects. So to counter that, there were solutions. The solution is you do a more real-time knockout by smash knock-in. So the smash knock-in provide a real-time tunable knockout of targets by inducer ASV. So then you can assess your phenotype in response to knockout in a more real-time manner. And without the inducer, the bulk tag is released because the protease function. So it's more advantageous than DTAG, which you have DTAG tag all the time, and it's cumbersome. It's going to block interactions with partners. So then smash tag remove that concern. So it's a more advantageous approach. So that's one of the caveats. The second one is CRISPR knockout. Some of the guys can have accidentally off-target effect. So that can be discovered, you don't have to do a whole genome sequence to find that. That takes time and cost. But when you do the recovery, the rescue, you can find that particular guide cannot be rescued sufficiently as the other guide phenotypically. So then that reason lies because that guide can accidentally trigger off-target effect
which can be validated by whole genome sequencing. So, those are two of the caveats. And I think the solution to the second one is you actually need multiple guides. So then you can figure out, which are the specific target effects that can be rescued, restored sufficiently by a rescue constructs, which are off target. That's one of the rare, more oddballs. Yeah, I think that's just, maybe partial of the answers, but, I welcome supplement from Dr. Wang. Thank you.
Baiqiu, would you like to add anything?
Yeah, I think Shelley pretty much covers it, the essential gene and some of the off-target effect. And for my presentation, the library screen, there's also a little bit concerned about that. If you use the general CRISPR knockout library, there's some essential gene that might be just missing because, you completely knock it out. But if you use a different approach, which is CRISPR-I, you just, inhibit some of the target, the gene expression. So basically, the gene expression is reduced, it's not completely gone. So in this way, you may identify some target that you would miss during the complete knockout library screen. So I think the message is very clear. There's definitely some shortcoming for this technology, but we also have solutions to address that.
Okay, thanks a lot, Baiqiu. Yes. So another situation that could arise is if your target is lethal in your cell of interest. What are the technologies that are available, perhaps instead of CRISPR, that you can use to identify those targets that are lethal?
Yeah, maybe I can address that. So as I mentioned in my presentation, for the knockdown study, you can do either constitutive or inducible. So in this case, you can apply inducible situation. So you can make a DOX-inducible knockdown cell line. So then you can have a tunable DOX-induced dose response of your knockdown that can correlate with your degree of phenotype change. So that can help conquer the lethality. You don't completely knock out, then you cannot generate the cell line. But if you have a DOX-inducible system, you can. The other option with CRISPR knockout, you can do DOX-inducible Cas9. So then you can have this cell line and you can put in DOX-induced CRISPR knockout and you can see lethality phenotype, but you still get this cell line working. And the other thing is that this cell line, once it is induced to have knockout, is actually irreversible from a genomic DNA level gene editing. So then, that's the endpoint of that cell line, but you can still, within the time window, do all the analysis that you need, all the proliferations, cell cycle, whatever biomarker study, you can manage that. So, these are the two options. For the DOX-inducible knockdown, withdrawal DOX, you still got the cell line maintained. So that's the difference.
I see. Okay, thank you, Shelley.
You're welcome.
Another question here in relation to artifacts that we might need to be aware of, particularly in modified cells. And do we need to look out for example, immune activation from an eliciting of phenotypic responses? Is this something, are there other artifacts that we need to look out for, and how do we protect ourselves against that?
Yeah, there might be a lot of artifacts depending on the target you're manipulating. So you really, when you talk about immune cells, that's in a context. So that's actually in the interacting context of your engineered cell line interacting with immune cells. So that's mostly in vivo context or in the co-culture system, right? So that's how you deal with the, when you have this target-mediated cell lines in an in vivo setting, you can look at TIL. You can look at what the immune cells they are. Have they changed populationally, functionally? You can do those assessments. You can use TIL assessment. You can do, all the immune cells assessment, typically biomarker changes, profiling, et cetera, It's just something that you can design and have a solution to find out. And from the other aspect, artifacts, I think one of the
artifact might be from off-target. So those are not specific true phenotype impact of your target manipulation, but rather artifact because of undesired off-target. So these needs to be sorted out. You can find it out by rescue studies. If it cannot be sufficient rescued, well, the other SH can, that means it is an off-target effect. You can do whole genome sequencing to find out. And if you really want to do that. And I think that's the other off-target and other artifacts. I think most of the other artifacts can also resulting from the target signaling pathways, how intertwined with the other pathways and impact on the other players. So that can be secondary effect. That's not a direct phenotype from your target knockout. So this needs to be based on a solid scholarship and understanding the pathway, the signaling, crosstalk, and then you can sort out, is it a true direct phenotype impact of your target manipulation or rather secondary? Is because of interference of other pathways. This is getting, can be very complicated, comprehensive, but I think all based on a very solid scholarship, you will find out. So that's how at HDB, we have a very strong scholarship troubleshooting capability. We can help you figure out what is the true effect, what might be artifact. Yeah, I think that's just from me. And if Baiqiu has more comment, please feel free to add on.
Yeah, I would just add on one point. I think Shelley made a very good point. Some of the artifact maybe just coming from, maybe not sufficient understanding of the biology. Other than that, I think from a technology point of view, we can also address that. For example, sometimes, it caused by just using one approach to attack the function analysis, but we have many options on our platform. You can just tackle this issue, from different angle, from RNA level, from protein level, or even completely change the methodology. I think that will also help from a technology point of view to confirm your result and to, either validate or devalidate the target.
I see. Let me just ask one final, quite specific question, and that's whether it's possible to have a complication through an immunomodulatory effect of the guide RNA itself.
I beg your pardon. Can you clarify question?
Sure. Let me ask quite specifically.
Yes.
Are there possible immunomodulatory effects of the guide RNA during the CRISPR experiment?
Yes, so that depends on the target you're manipulating. So some of the target, yes, they do have immunomodulatory. It has been reported in the literature. For example, Propelled, the CDK4 and 6 inhibitors, it can really boost the immune cells, like the CD8 effector T cells because of this manipulation of this target. This can be observed that reported in the Nature paper by different groups. You have boosted immune potency of the immune cells because of the increased antigen presentation. So there is an impact, very sophisticatedly involved, during this cancer cell immune cell dialogue. So you change the target on the cancer cells, eventually it's going to impact on its interaction with the immune cells. And some of the target manipulation can have kind of double roles. So it kills the cancer cells. On the other side, on a kind of a concentrated role, it boosted the immune cells cancer killing effect. So that is something, that has been observed, not in just in Propelled, this one CDK4/6 inhibitor, but in other cancer targets as well. So this is a very intriguing
and very interesting area that we can look at. And from our platform, we have the immune cells, cancer cell co-culture system, and we can provide, kind of solutions and design. We can help to look at those. How, for example, from the macrophages, ADCP, if you manipulate a player that can impact on the don't eat me CD48 surface markers, you can show an impact on the ADCP function of the macrophages. So you can design this co-culture macrophages with the target cancer line. And through this CRISPR knockout of the target, you can find increased ADCP, the macrophage engulfing cancer cells more evidently. So, this is something, maybe, help address your immunomodulator. So it's modulate immune cells functions. And also you can assess that, by all kind of immune assay format that we have here.
Perfect. Thank you very much, Shelley. I hope that answers the question, but if any of the guests would like to have any follow-up questions, please do feel free to email us through the contact details that were provided. I think we're out of time now, but I'd like to thank both of you for taking the time to discuss your respective platforms with us and our guests today. And thanks very much to the LRIG team for their great support and organization. I do hope our guests enjoy the rest of the conference, and please do feel free to reach out for us for any further information. Shelley, Baichu, thank you very much again for joining us.
Thank you.
Thank you. Bye.
Bye-bye.
Bye-bye.